All right. Okay, so today's lecture we are going to look at a new chapter, which is chapter 4. We finished chapter 3 last week. So we are going to look at chapter 4, which is on enzymes. Okay, so these are the subtopics of this chapter. So there's quite a lot of things that you need to understand for this chapter. Although the notes is not so much, and there's about I think 40 plus slides or 50 something like that. Okay. So these are the subtopic. The first one, catalysis and concept of activation energy. So you have to know what is activation energy, what impact does it gives to the reaction, and how the enzyme involves in lowering the activation energy of certain reaction. And then second, mechanism of enzyme actions. Okay, so we know that enzyme is a molecule, protein molecule that is to catalyze reactions. Okay, inside cells. So in order for the enzyme to catalyze reaction, it has to have active site for substrate to bind and reaction to occur to produce products. Okay, so the way on how the substrate bind to the enzymes will explain on how the enzyme will work. Okay, um, so that is mechanism of enzyme action. And then factors affecting the rate of enzyme reaction. So we know that enzymes are protein molecule. Okay, so one of the factor that may influence the rate of reactions is, for example, temperature. Okay, so as you increase temperature of the surrounding, the rate of reaction increases because as you increase the temperature, it increases the collision between substrate and also enzyme, and so therefore increase the rate of reaction, but up until a certain point. Kan? So up until the optimum temperature. Kan? So optimum temperature has the highest rate of reaction, but beyond that optimum temperature, due to the high temperature, it can cause the enzyme to denature. So we know that enzymes are protein. So the increase in temperature may disrupt, for example, hydrogen bonds between the amino acids in the polypeptide chain. This one we have looked at during chapter 1, right? Okay, in the protein subtopic. We know that enzymes are protein. Protein are made up of amino acids and then attached together to form polymers of amino acid, which is polypeptide. Kan? So those polypeptide will be arranged in certain structure through the interaction between side chains, such as the interaction between side chain through hydrogen bonds. Okay, so if you increase temperature, the hydrogen bond that hold the structure of the protein will be broken, for example, and the protein loses its shape. Enzymes, okay, it is protein in the globular shape, globular protein. Okay? So folded into specific shape so that the enzyme can function. So if you increase temperature, you can disrupt the structure of the protein. Okay, and then another factor is by changing the pH of the surrounding. Kan? So certain enzyme will work best, for example, in acidic condition or in neutral pH or in basic condition. It depends on the type of enzymes. Okay, and then we have the next one, enzyme kinetics. Kan? So enzyme kinetics, this one we are going to look at graphs that shows you rate of reaction and the factor that influence the rate of reaction, enzyme kinetics. Okay, and then cofactors. So cofactors, for example, are molecules or ions that aid in the enzyme reaction. Okay, so the cofactors, for example, will sit or bind in the active site of the enzyme, can cause the enzyme, for example, to be activated. So once the enzyme is activated, so the reaction can occur. Kan? So cofactors, it aid in the enzyme in it helps the enzyme reactions. Okay, to catalyze chemical reactions. And then the next one is enzyme inhibition. Okay, enzyme inhibition. So you know that, okay, enzyme, it has active site. Okay, so active site is where substrate will bind. So apart from having active site, the enzyme also have another site. We call it as allosteric site. Okay, so allosteric site is where, for example, molecule will bind, either it can activate the enzyme or inhibit enzymes. Okay, so that molecule we call it as inhibitor. So this inhibitor can either bind to the active site to prevent substrate from binding to the active site and reaction cannot occur, or it binds to the allosteric site. It changes the shape of the enzyme and causes substrate cannot bind to the active site of the enzyme and reaction cannot occur. Okay, so there's two sites where the inhibitor can bind, in the active site or in the allosteric site. In the active site, prevents directly substrate from binding. In the allosteric site, it causes the enzyme to change shape. Okay, so therefore, the substrate cannot enter, no reaction can occur. Okay, so that is enzyme inhibition. Okay, part of it. And then 4.7, regulation of enzyme activity. So we are going to look at what are the factors that regulate enzyme activities, and then how enzyme are classified based on the type of reaction the enzyme catalyzes. So for example, in chapter 1, can we looked at the process of uh, hydrolysis, kan? So hydrolysis where in a polymer that consists of many monomers, kan? So for example, the monomers can be monosaccharides in polysaccharides, or amino acid in polypeptide, for example. So in order for you to break the bond between the monomers is through the process of hydrolysis with the aid of enzyme in the reaction, kan? So because the enzyme involved in the process of hydrolysis to break bond with the aid of water, so that group of enzyme we call it as hydrolase. Hydrolase reaction is hydrolysis. The enzyme is hydrolase. Okay. So usually the name of enzyme, we know that it is enzyme. Usually at the end it has the word, it has the ending "ase", kan, suffix "ase" at the back, kan. But okay, usually. So in another reaction, for the reaction that involves redox reaction, redox reaction, the transfer of electron from one molecule to another molecule, the process of redox reaction, oxidation reduction reaction. So that group of enzyme that involves in redox reaction, we call it as oxidase, oxidase, all right? So there are 6 classes of enzyme, if I'm not mistaken, based on the type of reaction it catalyzes. And then last part is enzyme technology. Okay, so enzyme technology. So for that part, we are going to look at the application of enzyme in industry, for example, just on the surface. Okay, touching on the surface, not so detailed, right? So these are the learning outcomes. For the first, these are the explanation, the learning outcome based on that subtopic. Explain how an enzyme lowers activation energy of a reaction. Explain the hypothesis related to the mechanism of enzyme action. There's 2 hypotheses, lock and key and induced fit mechanism. And then explain the formation of enzyme substrate complex. Explain factors that affects enzyme reactions. And then explain the models of enzyme kinetics, and then explain the double reciprocal plots for enzyme kinetics. For this one, there is graph of which you have to understand. Okay. And then this type of cofactors, discuss the mechanism of enzyme inhibition, explain allosteric site in an enzyme, okay? Discuss how enzyme inhibitors and activators regulate enzyme activity in a cell. So enzyme inhibitor or activators are a molecule that will bind to allosteric site and causes the enzyme to change shape either in the active or inactive form. And then list the classes of enzyme and explain their role, and then discuss the importance and the main techniques of immobilized enzymes. Okay, so so first of all, you need to know the definition, what is enzymes. Okay, so enzyme, based on the glossary in the Solomon book, if you have Solomon with you, so this is in the glossary part. You have to know how to use your reference book. Okay, so you have your reference book, make sure that you fully utilize the book. Okay, so at the back there's glossary. So any terms that you don't know the definition, you can look up in the glossary. Yeah, so based on that, you search for the word enzyme. So this is the definition given in the Solomon. Every book's own definition, but the concept or the concept is the same, which is molecules, organic molecules that involves in reactions, specific reactions. Okay, so an organic catalyst, usually a protein that increase the rate of reaction. Okay, uh, of specific reaction by lowering the activation energy required for that reaction. Usually for a reaction to occur, okay, for substrate to be converted into product, it requires some form of energy. Yeah, so some form of energy. So this is what we call it as activation energy. So for a reaction that, for example, that doesn't involve enzyme, so the reaction can still occur, but it will take a long time for the reaction to occur because the system need to absorb the energy from the surrounding in order for the substrate to be converted into the product. Okay, by having enzyme in the reaction, this will speed up the rate of the reaction, accelerate the rate of reaction by lowering the activation energy of that reaction. Okay, so reaction still boleh berlaku kalau tak ada enzyme tapi lambat. Sebab apa? Nak kena absorb energy from the surrounding. Kan? The substrate needs to absorb energy from the surrounding so that the substrate can be converted into product. Okay. So these are the keywords that you need to know on enzyme. Speed up the rate of reaction and lower activation energy by lowering activation energy for that reaction. Okay, so these are the characteristics of enzyme. We know that it is globular protein. What are globular protein? You have a polypeptide chain that fold into specific shapes due to the interaction of side chain, right? So this is basically to form the globular protein structure. It is at the tertiary level, usually at the tertiary level due to the interaction between side chain through hydrogen bonding or disulfide bridge or ionic bonds, hydrophobic interaction, van der Waals interaction. So the interaction between side chain causes the polypeptide chain to be folded into specific shape to produce globular protein. Okay, so that's why earlier I said if you increase temperature, for example, so the interaction between the side chain through hydrogen bonding, for example, so it breaks the hydrogen bond and the polypeptide chain loses its functional shape. Okay, so tak adalah active site nak ada kat situ, so substrate cannot bind once the protein has been denatured. Okay, so and then act as catalyst, kan? Catalyst to catalyze the chemical reaction and then not permanently altered or changed, kan? So the enzyme is just to help the reaction to speed up, kan? So the enzyme will not change shape and will not be consumed by the reaction, kan? So it will not be used or converted into another form. Okay, and then can be reused over and over again, right? Okay, so can be used for the next reaction. Okay, so have one or more active sites where substrates bind to. So usually we know that enzyme has active site. So an enzyme, it can have one or more active site. So when it has more active site, it can bind to more substrate and speed up the rate of reaction, can accelerate rate of reaction, and then lowers the activation energy that is required for the reaction to occur. Okay, so next slide. Okay, so this is the on the activation energy, catalysis and concept of activation energy. So activation energy, this is the symbol EA, is subscript, also known as energy of activation. So it is the initial, so this is the definition. Okay, it is the initial energy needed to start a chemical reactions. Okay, so source of the activation energy is heat from the surrounding. Okay, so heat from the surrounding. So the substrate needs to absorb heat from the surrounding in order, for example, to vibrate for example, the structure of the substrate molecule and causes maybe bonds within the molecule to break, then only the substrate can be converted into products. So if the source of energy comes only from the surrounding, so it takes time for the reaction to occur. So that's why reaction needs enzyme to speed up the rate of reaction. Instead of substrate depends on the heat from the surrounding, the having enzyme in the reaction can accelerate the rate of reaction. Okay. Okay, so how does enzyme lowers activation energy? Okay, enzyme lowers the activation energy by one, bringing the substrate together in the correct orientation. Okay, so one reaction may require several substrates to be reacted upon each other. So they need to sit properly. Okay, so they need to sit properly so that the reaction can occur. Kalau dia bergerak-gerak-gerak, macam mana dia nak reaction tu nak berlaku kan? So you have the active site of the enzyme, active site of the enzyme. So the substrate needs to sit properly together in the correct orientation so that reaction can occur. Okay, so provide a favorable cellular microenvironment. Okay? So it provides cellular microenvironment that is to aid in the enzymatic reaction. Okay, so and then the third one, stretching the bond of substrate towards their transition state. And so once the substrate bind to the active site, it causes maybe bonds inside the substrate to vibrate, and then before the bond will be broken, so that is transition state. So it causes the molecule of the substrate to transition state is before substrate converted into product. Okay, and then forming temporary covalent bond with substrate. So enzyme can form temporary bond, covalent bond with the substrate. So this is how enzyme lowers the activation energy of a reaction. Okay, so this graph it shows you the rate of reaction for reaction that involves enzyme and also for the reaction that does not involve enzyme. Okay, so first we look at the black graph here. Okay, so initially we have the reactants. Okay, so this is the reactants. So it has two reactants in this case again. So the reactants will bind to the for the reaction that does not involve enzymes, for example. So as you can see, the graph is a lot higher and takes a longer time. So why is the graph is a lot higher? Because the reactant, the substrate needs to absorb a lot of energy from the surrounding, a lot of energy to cause, for example, the bond within the substrate to vibrate. Okay, so in order for the bond to be broken. So this is the condition we call it as transition state before reactant is converted into product. Okay, so as you can see, the graph that does not involve any enzyme, the activation energy is a lot. Okay, a lot. Okay, higher amount of activation energy is required, and as you can see, the graph takes a longer time. Okay, a longer time for the reaction to occur for before the reactant in the transition state to be converted into product. Okay, so but for the red graph here is for the reaction that involves enzyme. So as you can see, due to the presence of enzyme, the activation energy is much more lower and the rate of reaction is much more faster. Okay, before it is converted into product. We'll take a break first for the azan. Okay, so this graph you basically for this graph you have to know which one involves enzyme, which one does not involve enzyme. Okay, the one that involve enzyme would have lower activation energy and the rate of reaction is much more faster, accelerates the rate of reaction. Okay. And then another thing that you have to take note based on this graph is this delta G. Okay, delta G is the change in free energy with or without enzyme. The delta G value is the same. Okay. Uh, it says here for point number 3, an enzyme cannot change the delta G, the change in free energy. Instead, it speeds up a reaction that would eventually occur. Ada tak ada enzyme, reaction akan still berlaku, cuma cepat ke lambat. Macam tu lah. Okay. Okay. So next subtopic is on mechanism of enzyme action. Okay, so the first, so for this one, there's two hypotheses that you have to understand. Okay, the first one is lock and key model. Okay, so lock and key model. The second one is induced fit model. Okay, so mechanism how enzyme reaction occurs. Okay, so lock and key kan. So if you look at this analogy, we have here the lock and also the key. So the lock mangga lah kan, mangga yang mana you shove your key into. Okay, so the lock will be fixed in shape so that it can only receive specific key kan. So tak boleh lah guna kunci pagar pintu rumah awak yang kat luar dengan pintu pagar kat belakang kan. So they have different shapes kan. So the shape of the active site is fixed to receive specific substrate to bind to the active site. Okay. So the substrate is complementary to the active site of the enzyme. Okay, so the shape of the substrate complements the shape of the active site. If you use the word the substrate is the same, the shape of the substrate is the same as the active site, it is wrong. It is not the same. Okay, it is not the same. So it is complementary. This saling complement lah. Okay, complementary. Okay. So do not use the word same. So dia bagi definition yang salah kat situ. Okay. So the substrate is complementary to the active site of the enzyme. So the substrate bind to the active site of enzyme to form enzyme substrate complex. Okay, enzyme substrate complex. So this is enzyme substrate complex. Okay. So and then when this occurs, the formation of enzyme substrate complex, what happens? Bonds will be broken. Okay, a breakage of all bonds and formation of new bonds. Okay, so the substrate will then convert it into product. The product will obviously have a different shape from the substrate. So now the product, because of its difference in the shape compared to the initial substrate, so the product will be released. Okay, released from the enzyme. So it says here this mechanism is now considered as incorrect. So this is just a theory hypothesis, kan? Just a hypothesis to explain on how actually enzyme works, kan? But actually through many research by many scientists, so for the enzyme to work, it is more it follows more towards the induced fit model. Okay. Okay, so for this induced fit model, it says saya lupa nak highlight. Okay, the words here can usually these are the keywords that you need to take note kan. So substrate is complementary to the active site to form binds to the active site to form enzyme substrate complex. Then this occur, product form, different shape and release from the enzyme. So these are the keywords that you need to take note. Okay. Okay, so next, for the next one is induced fit model. Okay, so substrate is not complementary. Okay, substrate is not complementary to the active site of an enzyme. So as you can see, so this is the enzyme with the active site, and then this is the substrate. The substrate shapes does not exactly fit into the shape of the active site. Okay, but as the substrate binds into the active site, it causes the active site to change shape slightly. It induces a slight conformational change in the shape of the active site. Okay, so asalnya maybe, so this is the substrate, maybe this is the active site, macam tu kan. So not exactly complementary. So but as the substrate enters, the active site will change slightly to fit with the shape of the substrate. Okay, so induces a slight change, a slight conformational change to the active site of enzyme. The shape of the substrate also changes slightly, and then catalytic groups on the enzyme are in the correct orientation with the substrate. Okay, so and then if I'm not mistaken, catalytic groups, we know enzyme, it is made up of amino acids, can amino acids, and then protruding from the amino acid, we have the side chain, for example. So based on the different types of side chain, it may be acidic side chain, basic side chain that has charges, for example. They may involve in the reactions, can. So catalytic groups on the enzymes, okay, are in the correct orientation with the substrate, and then causing bonds to be broken and new bonds to be formed. And then once the bonds is the bond between within the substrate. So the substrate will change into product. So the product will have a different shape from the substrate. Product released from the enzyme, and enzyme returns to its original shape. Okay, so if you go through your question in the workbook, okay, so these are the points that you need to take note. Okay, poin-poin ni sangat penting. Yang ini sama je macam yang tadi. Kan? Ini. So apa yang membezakan lock and key is this one. Okay. Not complementary induces a slight conformational change. And then once the product release, the active site returns to its original shape. So these are the points that you need to take note, okay, on induced fit model. Okay. So next, catalytic cycle of an enzymes. Okay, so this is it is this diagram to show you how the reaction occurs with enzymes. Okay, so the first one, substrate. So these are substrates. Okay, so substrates in this case it involves several substrates. So substrate enters the active site. Enzyme change shape. Change shape is in this fit. Okay, change shape such as that its active site enfolds the substrate, which is the induced fit model. Okay, and then substrate held in active site by weak interactions such as hydrogen bonds and also ionic bonds. Substrate held in active site by weak interactions such as hydrogen bonds and ionic bonds. This one, as I've mentioned earlier, okay, protein terdiri daripada amino acid. Tengok baca polypeptide chain, kan dia ada backbone dia ada repeating unit of nitrogen carbon carbon, right? Ni chapter 1 ya, orang nak test minggu depan. And then protruding from that backbone of polypeptide chain, there is the side chain, kan? So side chain ada 4 jenis, kan? Satu polar, and then non-polar, and then ionic side chains, electrically charged side chain, acidic or basic, kan? So due to the side chain of the amino acid along the polypeptide chain in the structure of the protein, it will interact with the substrate binding in the active site. Okay, so interaction can be through the formation of hydrogen bonds or ion bonds. Okay, are you? Okay, ada angkat tangan ke? Tak ada. Okay, so next, active site therefore can lower the activation energy and speed up the rate of reaction. So this one here is the, kat sini ada apa? Transition state, kan? Transition state before the reactant converted into the products, kan? So product will have a different shape from the active site and then therefore release from the active site, and then the enzyme is free again to catalyze the next reaction. Okay. Okay, so next we are going to look at factors that affects the rate of enzyme reactions. Okay, so the first one is enzyme concentrations. Okay, so the more that you add enzyme into the reaction, the rate of reaction increases, but up until certain point, because for enzyme to if you keep on increasing enzyme concentration, but you have limited substrate, obviously the rate of reaction will stop at certain point, kan? So the rate of reaction will reach constant rate of reaction. Okay, so that is enzyme concentration. The more that you add enzyme, the higher the rate of reaction, but up until certain point, because substrate is the limiting factor. You have limited amount of substrate. Okay, but if you have excess of substrate, kan? So as you increase as you increase enzyme concentration, and there's a lot of substrate, there's never ending substrate. For example, the rate of reaction will keep on increasing. Okay, so and then temperature, kan? So temperature, kalau enzyme concentration graph dia sama ada increase macam tu ataupun constant rate. For temperature, the graph will be like this, kan? Bentuk bukit macam tu, kan? So rate of reaction increase as you increase the temperature up until optimum rate of reaction. You have rate of reaction as its optimum rate, but as you increase the temperature even further, the graph will decrease, because as you increase the temperature, the increase in temperature will cause the enzyme to denature and slows down the rate of reaction, okay, until it stops. Okay, and then same goes to pH. Kan? So rate of reaction will increase as you fix the pH up until the optimum pH. So it depends on the enzyme lah. So other enzyme can work best in acidic condition, some enzyme in in neutral pH, or some enzyme in basic condition. So it depends on the type of enzyme, but same goes to the temperature. The graph produced is also graph yang bell shape tu. Okay, optimum pH with the maximum rate of reaction. Okay, and then substrate concentration, kan? So the graph macam sama lebih kurang macam enzyme, kan? So as you add more substrate to the reaction, rate of reaction will increase until it reach constant rate of reaction, because for this one, enzyme is the limiting factor, kan? So if you keep on adding substrate to the reaction, but if you have limited amount of enzymes, so the rate of reaction will reach constant rate of reaction. Okay, so let's look at the explanation, the same explanation. Okay, so the first factor, enzyme concentration. So this is the two graphs that will produce either straight like this, kan? What we call it as proportional. Kan? Rate of reaction is proportional. As you increase the enzyme concentration, but you have unlimited substrate, the rate of reaction will keep on increasing, because you have a lot of substrate, unlimited substrate. But if the substrate is limited, you only have certain amount of substrate added into the reaction. So as you increase the enzyme concentration, the rate of reaction increases, but up until a certain point where the reaction will reach constant rate of reaction, because substrate is the limiting factor. You have fixed amount of substrate. Okay, so it says here, if substrate is not the limiting factor, the red graph here, can you have a lot of substrate, unlimited, so the rate of reaction is proportional. As you increase the enzyme concentration, the rate of reaction will keep on increasing. Okay, but if the substrate is the limiting factor, the blue one here, adding more enzyme will not help to increase the rate of reaction, because you have only certain amount of substrate added into the reactions. Okay, so this is the point where the rate of reaction reach constant rate of reaction. Okay. Okay, so next one, the second factor is temperature. Okay, so it says here the rate of reaction increases with increasing temperature. Okay, so for this one, most human enzymes, okay, will work at its optimum temperature based on our body temperature, which is maintained at 37 degrees Celsius. So as you increase the rate, as the temperature increases up to until optimum temperature, so this is the maximum rate of reaction. But beyond this temperature, beyond 37, for example, the enzyme will start to denature and rate of reaction will decrease. So that's why kalau macam seseorang tu demam, kena make sure dia punya temperature tak go beyond 40 degrees Celsius actually. So kalau lebih pada tu, so you have to take action, especially if small kids kan, yang demam, you have to really monitor, kan. So you do not want, due to the high temperature, it affects the metabolism within cells, kan, the reaction within body cells. Okay. So so it says here, as temperature increases, what effect does it give? It causes the molecule, the substrate, to collide, increase the collision. Okay, increase the collision between the substrate, so that the substrate can bind to the active site. It increases the collision between substrate and to enter into the active site of enzyme. Okay, so the rate of reaction doubles for every 10 degrees rise in temperature. So every 10 degrees, the rate of reaction doubles. Okay. So it says here, at low temperature, maybe we can consider zero up until 20 degrees, kan, before 30 ni, enzyme activity is reduced, kan, or does not occur at all, especially if the temperature is really really low. Okay, so because of a lack of energy for the reaction to occur. Okay, and then optimum temperature enzymes are the most active, kan. So at optimum temperature enzyme is the condition where the enzyme is most active, rate of the reaction is the fastest, and balance between enzyme stability and kinetic energy. Yes, those enzyme will be in more stable condition, and there's more collision between enzyme and also active sites. Okay, sorry, enzyme and also the substrate. So and then above optimum temperature, so due to the high temperature, the kinetic energy is even more. Okay, so the high kinetic energy will disrupt the 3D structure of active site, especially this will break the hydrogen bonds between the side chain of amino acid along the polypeptide chain, and causes the enzyme to denature. The enzyme loses its functional shape. They are suddenly globular. So bila ada increase in temperature, the hydrogen bond, for example, will break due to the high kinetic energy and causes the enzyme to denature. Okay, so it depends on the organisms. Okay, so for humans, the optimum temperature is 37, but for other organisms, such as for bacteria that lives in water that are really hot, can hot spring. Okay, so this bacteria will, the enzyme in the bacteria will work at high temperature, kan. So maybe around 70 plus before 80. Depends on the type of bacteria lah. Kalau dia duduk kat area yang sangat panas in the hot spring, so this may be the optimum temperature. Okay. Okay, so the temperature coefficient, which is Q10, so this is the definition, is the factor by which the rate of reaction increases with every 10 degrees rise in temperature. So if you increase the temperature by 10, rate of reaction doubles, for example, kan. So if for example this is certain temperature, kan, and then you insert into this equation and gives a Q10 value of 2, for example, it means that the rate of reaction doubles. But if Q10 is 3, maksudnya the reaction triples, for example. Okay, so depends lah. So if Q10 is 2, the rate of reaction doubles. If Q10 is 3, the rate of reaction triples, for example. Okay, so if Q10 is 2, the rate of reaction doubles with every 10 degrees rise in temperature. Okay. So next, the third factor is pH. Kan? So optimum pH is the surrounding pH where enzyme is most active. Okay. So it depends on the type of enzyme. For example, here we have pepsin. Okay, so pepsin is the enzyme secreted by the stomach lining. Okay, so the stomach lining will secrete enzyme pepsin for the digestion of protein. Okay, so digestion of protein will occur, for example, in the stomach due to the release of pepsin. But pepsin, initially, when it is secreted by the lining of the stomach, it is secreted in the form of inactive form. We call that as pepsinogen. Okay, initially, the wall of the stomach secretes pepsinogen, okay, which is in the form of inactive, okay, enzyme. So in order to activate the enzyme, the pepsinogen, it has to have acidic condition. Okay. So in order to provide the acidic condition, so your stomach lining also will secrete another substance, okay, which is hydrochloric acids. Kan? So hydrochloric acid, which is secreted also by the stomach lining, will activate the pepsinogen and convert it into pepsin. So then only pepsin can catalyze the reaction. Okay, so this is the optimal pH for pepsin. It has to work at acidic condition, which is contributed by the hydrochloric acid to cause the pH to be acidic. Okay. And then another type of enzyme, for example, enzyme which is secreted by the intestinal wall. Kan? So for this type of enzyme, such as trypsin, it has to work in basic conditions. So acid is due to the presence of a lot of hydrogen ion. Basic is due to the presence of hydroxide or H negative ion ni. Okay. So changes in pH due to different concentration of H plus or H negative ions here. So it changes the ionic charges on the active site of enzyme. Again, this one you have to understand the concept of polypeptide tadi lah kan. So polypeptide, along polypeptide ada amino acid. In the tertiary structure of protein, side chain will interact with one another. Okay, so side chain of type acid, kan acid yang ada, contohnya ada carboxyl group, so dia akan ionize jadi COO negative, right? Okay. Bila dia jumpa dengan lagi satu amino acid yang ada amino group, basic, so basic dia akan jadi NH3 plus, negative and positive will be interacted to form ionic charges. Okay, but when you mess around with the pH, it causes this ionic bond to break, and the protein, the enzyme loses its original shape. Okay, so ini kita tengok balik yang konsep yang tertiary structure. Okay, tertiary structure. So changes ionic charges on the active site of enzyme, break weak bonds such as hydrogen bonds and ionic bonds between the side chain of amino acids along the polypeptide chain. So when the bonds broken, kan asalnya dalam bentuk globular, so the polypeptide chain will lose its shape lah. Okay, alters the 3D shape of the of the active site. So when the active site changes shape, substrate cannot enter into the active site, so reaction cannot occur. So less product produced or no product produced at all due to the enzyme that has been denatured. Okay. So next, substrate concentration. Okay. So for this one, for this graph, as you can see, it produces this type of graph. Okay, where it has 3 points. Okay, three points that you have to know what happened at these 3 points of the graph. Okay, so substrate concentration, x-axis is the one that you manipulate. Okay, so substrate concentration, you keep on increasing the substrate concentration to see the rate of reaction. Okay, so point A here. Okay, so this is the point. The rate of reaction increases rapidly, steeply. Okay, with increasing substrate concentration. So due to more collision between enzyme and substrate. Okay, for this one, you have in the system, in the reaction, so you have fixed amount of enzyme. Okay, so ada maybe ada 100 enzyme molecule kat situ. Okay, so initially during the reaction, 100 enzyme yang awak tambah slowly, okay, 10 enzyme, 10 substrate. 10 substrate akan bind freely lah kepada active site of the enzyme, and then you add another 20, and then you add another to 30, 40, 50 for example. And so the more that you add the substrate, the higher the rate of reaction because you have ample of enzyme at that point of time. Okay, but as you add on more substrate to the reaction, the rate of reaction increases but at a lower rate. Kan? So kita ada 100 tadi, so ada tambah sampai let's say 80 substrate kat situ kan, 80 for example. So yang baru nak masuk ni dia kena cakap kat mana yang free lagi, kat mana yang free lagi contohnya kan. So the rate of reaction will slow down. Okay. So rate of reaction increases but at a lower rate because fewer active sites are available. Okay. So and then point C, maximum rate of reaction, which is we call it as Vmax, maximum velocity here. This is the point where you can note there's Vmax, maximum rate of reaction, maximum velocity, because all active sites are saturated with substrate. Ada 100 enzyme, ada 100 substrate for example, ataupun tambah lagi 200 contohnya. Okay. So all the active site is now occupied with the substrate. So yang lain tu kena tunggu kejap. Faham tak? Kena tunggu kejap until the reaction completes for the next substrate to bind. So this is the rate, maximum rate of reaction kan. So constant rate, all active site are saturated with the substrate. Okay, so each point you have to take note lah what happened. So this one you have a lot of active site initially during the reaction. When you keep on increasing the substrate, there's still a lot of enzyme. But as you increase more substrate, active site will be fewer. Okay, so rate of reaction will slow down a bit until it reach constant rate of reaction where active site are saturated, okay, with the substrate. So far, do you have any questions? Ada soalan tak? Okay. Kalau tak ada, kita stop dulu ya. Boleh? Okay, so kita stop dulu our lecture today. If you have question, you can ask. If not, bolehlah kita bersulai. Okay, kelas?